In this video, we are going to discuss photoreception. Photoreception is the biochemical pathway in rods by which rods indicate the presence of light. First, let's look at the structure of a rod. A rod has two segments: an outer segment, which is composed of membranous discs, and an inner segment, where the cell organelles are located and most of the cell processes take place. Each rod synapses with a bipolar cell, which is a nerve cell. Neurotransmitters are released from the inner segment to the bipolar cell in the synaptic cleft. Like the neurons of your nervous system, the plasma membrane of the rod contains chemically gated sodium ion channels. These are located in the plasma membranes of those membranous discs, which are stacked like this in the outer segment. In these plasma discs, the chemically gated sodium channels are kept open through the presence of cyclic guanosine monophosphate. This chemical, also known as cGMP or cyclic GMP, is an energy molecule. Much like AMP is a derivative of ATP, GMP or cyclic GMP is a derivative of GTP. In the dark, cyclic GMP is present. When cyclic GMP is present, the membrane potential here is -40 millivolts. Now remember, in your nervous system, the resting potential is -65 millivolts. However, this process is quite different from that because an action potential is not created here. Instead, we are just creating an electric potential. At this resting potential, the inner segment is constantly releasing these neurotransmitters and also sending sodium up into the outer segment. When cyclic GMP is present, these sodium-gated channels are open, and so sodium flows freely out of this membrane into the cytoplasm. The movement of sodium is called the dark current. And this is quite easy to remember, because remember, this whole process is taking place in the dark, and rods give us ability to see in the dark. Thus, it's quite easy to remember. The outer segment's discs have a plasma membrane. Remember, a plasma membrane has that polar head and these non-polar tails, and embedded in this membrane is rhodopsin. Now remember, that is that pigment molecule that is critical to allowing us to perceive images. Within rhodopsin is a molecule called retinol. This is where the process of photoreception begins. So this is our rhodopsin molecule. Remember, this rhodopsin molecule is located in the outer segment of the rod. And that outer segment is where those membranous discs are located. And within those membranes, this rhodopsin is embedded. So what happens is that light enters, strikes the rhodopsin. When the rhodopsin is struck, this retinol isomerizes from the cis form to trans. And when this happens, this is our first step in which rhodopsin enters its active form and becomes known as meta-rhodopsin 2. Once it's in its active form, a chain of events occurs. What happens is that meta-rhodopsin activates transducin. Transducin is another molecule that can cause a chain reaction. Once transducin is active, it can in turn activate phosphodiesterase, which is another protein. Now, phosphodiesterase is important because it breaks down cyclic GMP to GMP. Now, if you remember, during the resting phase, we have our discs, and then they have those sodium-gated channels. You have all that cyclic GMP flowing, which allows the sodium to move out of the cell. So once there's no cyclic GMP, these channels are going to close. And once they close, then sodium is not going to be rushing out of the cell at all. Less sodium is going into the cytoplasm, so the dark current is reducing. That means that less neurotransmitters are being sent to that bipolar cell, slowing down the whole process once that cyclic GMP is gone. So once the dark current is reduced, the membrane potential hyperpolarizes. Remember that resting potential is negative 40 millivolts. Hyperpolarization means it goes below this to about negative 70 millivolts. Far below resting potential. Once this is complete, this means that a rod has absorbed a photon. After absorption, the rod needs to recover from this process. This recovery is known as bleaching. Retinol, as you remember, here, embedded in that outer membrane in those sacs, is in its trans form after the photoreception process is complete. So we need to return it from trans to cis. This process requires ATP. However, it takes time to get ATP, so in the meantime, there's a little bit of a delay before this rhodopsin is recovered. Usually, you don't notice when bleaching occurs, but if you've ever stared into a bright light or a camera flash for a long time, you'll see that ghost image after. This is because your rhodopsin molecules are still regenerating. During bleaching, metarhodopsin 2, that's that active form of rhodopsin, is transformed back to the inactive form, rhodopsin. This happens and cyclic GMP is regenerated, and that means that those chemically gated sodium channels in the outer membrane are reopened, and that means sodium can freely flow out of the cell again. Then the rod is ready to accept another photon of light. The purpose of this process, remember, is to indicate the presence of light.